So today I'm going to introduce to you the program called DNA Master and explain to you what this program does and help you set the program up for your phage genome annotation. So the specific learning goals are for you to understand the purpose of DNA Master and how the program functions, and be familiar with the programs within DNA Master, and finally be able to set the preferences in your DNA Master program so that you can successfully annotate your phage genome. So what are we starting with in this course? Well, last semester we left before break having received the 4 genome sequences for Sally Special, Cuke, T. Brady 12, and Shere Khan. And what I have here is part of the Sally Special genome. This is a FASTA sequence, which is a type of sequence file. And you can recognize a FASTA sequence by finding this little caret followed by a title or description of the sequence. Then there is a carriage return, followed by the actual nucleotide sequence of the genome. Now this genome is over 15,000 base pairs long, and so obviously I can't fit that on the screen, and I've just shown you just the very, very beginning of the sequence. Well, what we can do with this sequence as it is really not much, because it's just a string of letters to us and we don't know where the genes are. So we need to take this sequence and we can import it into the program DNA Master. And what DNA Master will do for us is predict where all the genes lie. And we can see this in a couple different ways. There's output that shows us the list of genes, what their starts are, and usually you can see what the stop is too. We can also see below this map that shows us where these genes lie and what orientation they are. These are all forward genes in green, and the red ones show us genes in the opposite orientation. So the phage genome is auto-annotated by DNA Master. And DNA Master can do this because it has several programs in it that analyze the DNA. And then these programs display the data from each of the different DNA analyses, and we can interpret the data, we can make modifications to the annotation as we see fit in DNA Master. So the 3 programs within DNA Master include 2 gene prediction programs. One is called Glimmer and one is called GeneMark. And we'll talk more specifically about these programs and how they work next week. But in any case, really briefly, what they do is they scan the genome and look for the largest open reading frames in the genome and determine what the codon usage pattern is in those open reading frames. And they determine the coding potential. And then based on those codon usage patterns and the coding potential, they determine where those patterns pop up in other places in the genome, and that's what helps them decide where the genes are in the genome. Now, both of these do the same thing, but they're using different algorithms. So sometimes Glimmer and GeneMark will agree on gene prediction, and sometimes they predict genes differently. The third program is tRNA prediction program called Aragon. So we begin with the FASTA file, which you download from PhagesDB. This is imported into DNA Master. And again, there's the output in the form of the list of genes and the starts and links of these genes. For each gene, you could click on it, and then you can see where the start is, where the stop is, how long the gene is. You can also see what the actual sequence of the gene is, what the sequence of the amino acid is, and there's a lot of other information linked to each gene to help us make decisions about whether or not we agree with these gene predictions. So this is called the features table, and we'll talk a little bit more about the features table shortly here. So why do we need to annotate the genome if DNA Master does it automatically? And the reason why is because the auto annotation is not always correct. There are genes that these programs sometimes predict, and then we look at it and later decide that's not a gene, or sometimes the auto annotation misses genes, and we have to detect them manually. The other reason is the starts for the genes are not always correct. And so we need to examine evidence for every single gene and decide whether or not we think the program has predicted the correct start. And finally, these programs cannot predict the function of the genes, and so we have to look at multiple pieces of information to determine whether we can predict a reasonable function or likely function for each gene. So this is output from DNA Master, and it's showing, you guys know that there are 6 possible frames for any given sequence, and I'm actually only showing 4 of the frames here because I didn't have enough room on the slide, and this is showing the first gene predicted in Sally Special and the second gene predicted in Sally Special. And if you look at this line that goes from top to bottom in this frame, that represents a stop of a upstream open reading frame. And you can see that's a very short open reading frame right here. And it's probably not a gene. These lines that are halfway through represent starts in that particular reading frame. So here's the first open reading frame for this gene. This would be the start of that gene. There's also this potential start and 2 potential starts here. Here is, in green, is the second predicted gene in Sally Special, and you can see that Glimmer has predicted this start as the correct start for the gene. But, and I haven't pointed to all of them, there are many, many other options here for potential starts. And our job is to determine whether Glimmer is correct. Sometimes GeneMark predicts a different start. And sometimes the correct start is not the start predicted by either Glimmer or GeneMark. And we have to determine which one is most likely the start. And again, like I said before, the computers can also not determine the function. So we have a lot of work to do, but these programs give us a big head start in terms of shaping the genome annotation. So there's 2 ways that information are stored in DNA Master. Information about the annotation is stored in the feature tables. This is the place where we'll spend the majority of our time, where we can see the data. It's easy to navigate. We can click on all these different tabs and find out different information about each gene, where it starts, where it stops, the length. We can take notes here. And we can also, as we see fit, change the annotation. Perhaps we don't agree with the 128 start, and we might change it to a different start. We can also add in functions that we think belong to each of these genes. So all this information and all the changes we make in our annotation are stored in the features table. There's the second place where information is stored separately is in documentation. And this is really just text information. It's the same information, but just in text, and it's a little bit more difficult to navigate, to read, to understand, and it's not a place where we would make changes in the annotation. So let's talk about how the information in these 2 different places interact. So we import the FASTA sequence of the genome, and that DNA sequence is analyzed by the different programs, and the output from those different programs is stored in documentation. And then, since it's not easy for us to understand the information in documentation, it is parsed into the feature table. And so all the information that was originally in the documentation ends up in the feature table, and this is what we see right after auto annotation, and this is where we can analyze and make changes. Now, there's a button in the documentation tab called Recreate, and the Recreate button allows us, as we make changes in the information in the features table, to rewrite the documentation so that the documentation matches the annotation that we're creating in the features table. Now there's also this button called Parse, and this Parse button you want to avoid pushing unless I say otherwise, and I can't think of any situations yet where you would want to do that because I haven't pushed it yet. But the reason why we want to avoid Parse is because it takes the contents from the documentation here and it would overwrite what's written in the features table. So by parsing, we're erasing everything in the features table and rewriting with whatever's in the documentation. Now this would be problematic if you're making changes here and storing them in the features table, but then you parse what's in the documentation. It's likely that you will lose all the changes you've made in your annotation. So you want to avoid the parse button and stick to just pushing recreate whenever it so pleases you, because that will constantly update the documentation information. So that's where I'm going to leave this video for now, and in our next video we will set up the preferences in DNA Master so that you can successfully annotate your genome.